Living things share a molecular composition that's fundamentally distinct from non-living matter, a key characteristic that sets the stage for our exploration of human biology. Today, we're diving into the essential features of life and the scientific process, drawing insights from the comprehensive Human Biology, Science, and Society lecture presentation developed by Professor Vanessa. When we consider what defines a living organism, several crucial characteristics come to mind. Living things have a different molecular makeup compared to non-living entities. They require energy and raw materials to sustain themselves, and they're all composed of cells, the basic units of life. Furthermore, living organisms maintain homeostasis, a stable internal environment, and respond to changes in their external surroundings. They also grow and reproduce, ensuring the continuation of their species. Perhaps most profoundly, populations of living things evolve over time, adapting to their environments. Now, based on these characteristics, living things are grouped into a classification system. We recognize three fundamental domains of life: domain bacteria, domain archaea, and domain eukarya. Within domain eukarya, we find four distinct kingdoms: kingdom protista, kingdom animalia, kingdom fungi, and kingdom plantae. The fundamental criteria used for this classification largely revolve around a few key features. First, the presence or absence of a nucleus. Domain bacteria and archaea are prokaryotes, meaning they lack a membrane-bound nucleus. Domain eukarya, on the other hand, consists of eukaryotes, all of which possess a membrane-bound nucleus. Second, the number of cells is important. Organisms can be either unicellular, composed of a single cell, or multicellular, made up of many cells. And finally, the type of metabolism they employ, how they acquire and use energy, helps distinguish them. Let's take a closer look at the four kingdoms within domain eukarya. Protista includes organisms that can be unicellular or simple multicellular, like protozoa, algae, and slime molds. Kingdom plantae consists of multicellular eukaryotic photosynthetic organisms, which produce their own food. Kingdom animalia comprises multicellular eukaryotic heterotrophic organisms, meaning they obtain nutrients by consuming other organisms. And finally, kingdom fungi are eukaryotic decomposers, including molds, yeasts, and mushrooms. To bring this classification home, let's consider humans. Our classification is as follows: domain eukarya, kingdom animalia, phylum chordata, class mammalia, order primates, family Hominidae, genus Homo, and species Sapiens. The smallest unit of this classification system is the species, defined as one or more populations of organisms with similar physical and functional characteristics that can interbreed and produce fertile offspring. The second smallest unit is the genus. Importantly, all living human beings belong to the same genus and species, which is Homo sapiens. Beyond this classification, humans possess several defining features that set us apart. We exhibit bipedalism, the ability to stand upright and walk on two legs. We have a relatively large brain compared to our body size, which contributes to our capacity for complex language, both spoken and written. And, uniquely, we have opposable thumbs, allowing us to grasp objects between the thumb and the tips of our fingers, which is crucial for tool use and fine motor skills. For instance, the way humans and chimpanzees hold small objects highlights this difference. Human biology can be studied at various levels of organization, starting from the most fundamental. It begins with atoms, which form molecules. Molecules combine to create cells, and cells organize into tissues. Tissues then form organs, and multiple organs work together in organ systems. These systems make up an organism, which exists within a population. Populations form communities, which interact within an ecosystem, and finally, all ecosystems together comprise the biosphere. Each of these levels presents its own unique issues and controversies for scientific investigation. Now let's shift our focus to science itself. Science is fundamentally the study of the natural world. It's two things simultaneously. It's a body of knowledge we've accumulated about the natural world, and it's also the rigorous process we use to acquire that knowledge. This process is what we call the scientific method. The scientific method is a systematic process for testing ideas, involving several key steps. First, we observe and generalize. This involves inductive reasoning, where we make generalizations based on specific observations about the world. For example, if every winter in the past was colder than the preceding summer, we might generalize that winter will always be colder than summer. The next step is to formulate a hypothesis. A hypothesis is a tentative statement about the natural world, a proposed explanation that can lead to testable deductions. An example might be, drug X would be an effective treatment for high blood pressure. Following this, we make a testable prediction. Hypotheses should be tested under many different conditions. These predictions should be based on the hypothesis itself and employ deductive reasoning, often taking the form of if-then statements. They need to be specific to be properly testable. So, building on our example, if drug X is an effective treatment for high blood pressure in humans, then a specified dosage of drug X will lower blood pressure in people with high blood pressure within 1 month. The fourth step is to experiment and observe. An experiment is a carefully planned and executed procedure designed to test a prediction based on a hypothesis. We then observe the results. After this, we modify the hypothesis as necessary and repeat the process of prediction and experimentation. If a prediction turns out to be false, we must modify the original hypothesis. If a prediction proves true, it only means one small part of the hypothesis has been supported, and further testing is always required. It's crucial to remember that hypotheses can never be proven absolutely true. They can only be supported or disproved through evidence. Designing and conducting an experiment requires careful consideration of variables. The independent variable is the component of the system that the researcher intentionally manipulates or changes. We also call this the manipulated variable. The dependent variable, on the other hand, is the observed change or response in the system that's caused by the manipulation of the independent variable. This is known as the responding variable. Let's apply this to our drug X example. To conduct a controlled experiment testing if drug X can reduce blood pressure, a large number of subjects with high blood pressure would be randomly divided into two groups. The experimental group receives drug X, while the control group receives a placebo. It's vital that all other variables that might affect the outcome, such as age, gender, previous health problems, and initial blood pressure, are equalized between the two groups. In this scenario, the receipt of either drug X or the placebo is the independent variable. The blood pressure of all participants, which is our dependent variable, is measured after 1 month and compared to their initial blood pressure. Once findings are obtained, it's essential to make them known, typically by disseminating them in peer-reviewed journals. Experts in the field rigorously approve these articles before publication, meaning the most accurate scientific information is usually found here. Results are considered valid only for the specific conditions under which an experiment was done. Complete documentation is also critical, as it enables other scientists to repeat experiments and develop and test their own predictions, ensuring the replicability and reliability of scientific knowledge. A well-tested hypothesis can eventually become a theory. For this to happen, a hypothesis must be broad in scope, extensively tested, supported over a significant period, explain a wide range of facts, and possess a high degree of reliability. Examples of well-established scientific theories include the theory of evolution and cell theory. However, even theories may be refined or even refuted in the future as new and better information emerges through continued scientific inquiry. It's also important to consider the sources of scientific information as they vary widely in style and quality. More reliable sources typically include peer-reviewed journals, science magazines, and credible nonfiction books. General interest news magazines and daily newspapers can also be reliable, though they may simplify complex topics. News websites on the internet also fall into this category. Less reliable sources, though their quality can vary, often include social media platforms like Facebook, Twitter, and Instagram, where information may not be vetted. When assessing the quality of an internet source, ask critical questions. Who authored the article or site? Do they have academic or professional credentials? Are they an expert in the field, or are they writing outside their expertise? What is the purpose of the website? Is it to provide objective information or to promote an agenda or product? When was the information updated? Is it current? And crucially, where is the information coming from? Are references provided to support the claims?
Learning to be a critical thinker means becoming a skeptic, questioning what you encounter. It involves learning how to read and interpret graphs accurately and appreciating the value of statistics. It also requires distinguishing between anecdotes, which are personal stories, and scientific evidence, which is systematically gathered. You must learn to separate facts from conclusions drawn from those facts and understand the crucial difference between correlation and causation. Just because two things happen together doesn't mean one causes the other. Ultimately, science plays a vital role in society. It continuously improves technology and the human condition, from medical advancements to understanding our planet. However, science also has its limits. It cannot answer every question, especially those beyond the natural world. Nevertheless, science helps us to make informed choices, equipping us with the knowledge to navigate complex issues in our lives and communities. Ultimately, understanding science as both a body of knowledge and a rigorous process empowers us to make informed choices and critically evaluate the vast amount of information we encounter daily. This deep dive into the characteristics of life and the scientific method was based on the insightful Human Biology, Science, and Society lecture presentation by Professor Vanessa. If you found this discussion valuable, please consider sharing the episode with someone who might appreciate learning more about these foundational biological concepts.